The Morality of Genetics by Corinne Manley. How prevalent are genetic mutations? There are approximately 25,000 known genes in the human genome, 3,000 of which could result in mutations linked to disease, and there are possibly more that have yet to be discovered. What are some of the best possible solutions? There is gene therapy, RNA inference, DNA double-strand breaks, which can be paired with non-homologous end joining or homology direct repair, and then there is RNA editing as well. What can we do with gene therapy? Gene therapy can be used to restore missing gene functions, transfer DNA or medicinal products to different types of cells in the body, and then the use of medication can be used to replace faulty DNA. These medications can act as substitutes where DNA is lacking or dysfunctional. One example of a genetic mutation, called phenylketonuria, or PKU for short, involves a genetic mutation of the phenylalanine hydroxylase enzyme. Also known as the PAH enzyme, is either missing or does not work, so the liver cannot process the amino acid phenylalanine. A person with PKU must avoid protein unless using gene therapy, and failure to comply with a strict diet will result in brain cell death. There are two options for phenylketonurics for gene therapy. The first one is called sapropterin dihydrochloride, most commonly known as Kuvan, which is an oral powder. It tells the PAH enzyme to start working, basically, and a person with PKU can now eat more, usually double, the amount of protein with a better functioning enzyme. The PKU patient still is on a monitored diet with a protein restriction, but there are some issues with this medication. If there is no PAH enzyme in the liver present, then there's nothing for the medication to communicate with, so this can only help to a certain extent. The second therapy option is called pegvaliase, which is most commonly known as Palynziq. This is a subcutaneous injection that attaches to excess phenylalanine within the body and allows it to be excreted through normal body processes. A person with PKU no longer needs to be on a diet with this medication, but there are also some issues with this one as well. The most notable issues include injection site reactions such as swelling, redness, bruising, and other issues. And there's also a possibility of anaphylaxis, so a person with this medication would need to carry EpiPens with them. And as it is not ethical to test on pregnant women during the trial phases, pregnant women have to be taken off of this medication during the course of their pregnancy. Another option is called RNA inference, which blocks the function of certain mutated genes that code for things like cancer and macular degeneration. RNA inference inhibits these genes from progressing to later stages. It blocks the genes from their full expression, but there are issues with RNA inference as well, such as it doesn't always completely suppress gene function or expression. So this will not work where complete suppression of a gene is needed for success. One technology used to enable genome editing starts with DNA double-strand breaks, or DSB. DNA double-strand break paired with either a non-homologous end joining or homology direct repair, and it starts with DSB being inserted into cells and then uses non-homologous end joining or homology direct repair to damage made at the insertion site. NHEJ works similarly to RNA inference. However, it can result in genes being permanently inactive or cause other mutations involving the loss of function of these cells. HDR could be used to replace mutated genes and restore function, and it is also more specific, which means it can be used to create a certain outcome for specific genes. But overall, these technologies cause more issues than gene therapy and RNA inference. In vitro editing involves the editing of separate messenger RNAs, and since it is in vitro, it is only done under lab supervision. It is not practical to practice on people, but it could be used to modify newly fertilized zygotes with a known family history of a particular disease. Further research is needed, however. Some issues with genome editing and therapy are that you can't get consent from everyone. This is a very unpredictable thing with very unpredictable consequences, and it can completely change nature versus nurture and the outlook for humans as an entire species. However, there are some arguments that this decision is no different than taking away rights or giving us rights, and it can be very similar to the Wade versus Roe topic going around about abortion right now. So, here's the real question. Is it ethical to keep trying? There was a survey done with 1,000 respondents in 10 countries, and this survey asked 4 questions. Should genome editing be done for therapeutic reasons? Should it be done in adults or prenatal? And then should gene editing be done for personal enhancements in adults or prenatal? The third question asked, would you have made the same decision if it were you in their shoes? And this was referring to very specific circumstances, such as something happening to a family member. And as for results, the respondents had a higher score of moral acceptance aimed at therapy over enhancement overall. Most said that they would have made the same choice if placed in their shoes. Prenatal had lower acceptance than adult, and most said that they would not have made the same choice if placed in their shoes, but it is still debatable. There were also some interviews conducted with 3 questions for each person, which included, do you support or disagree with gene editing and why? Are there any circumstances where you might want this technology used, and do you believe it is moral or ethical and why? And just a few of the answers were, for question 1, if it's a matter of curing something like cancer, then I think it should be used, but not to become the best athlete or something. For question 2, my sister actually has a disease that changed how our family had to handle everything. She needs a lot more attention. Her medical bills are where most of the family's income goes. So if she could be helped, I think it would improve a lot of people's quality of life. And then someone answered for question 3, it's not ethical at all. Just like the topics going around about abortion, it's not any more ethical to kill a baby than it is to change a baby before they're born. They don't have the ability to consent yet. Overall, most methods of gene editing can only be utilized or tested for shortly after conception or early into a pregnancy. Several options, including changing messenger RNA or RNA inference, which both need further study to determine effectiveness. Then there are some methods approved for, such as Palynziq and Kuvan, used for phenylketonuric patients. Many statements aimed at supporting gene therapy and editing, but just as many were opposing gene therapy and editing. And thank you for listening to my presentation.